tryptophan operon, which contains 5 structural genes, that is E, D, C, B, and A, and that code for enzymes required for tryptophan synthesis. Regulatory elements are in tryptophan operon are regulatory gene, promoter, operator, attenuator, and terminator. The transcription of tryptophan operon is controlled by two mechanisms, that is operator control and attenuation control. And this tryptophan operon is always in on mode. Operator control. When the tryptophan is in plenty, two molecules of tryptophan binds to a repressor protein and causes it to block to operator site, and which prevents the messenger RNA transcription and expression of structural genes is blocked. So the tryptophan acts as a co-repressor, and this control is autoregulation or feedback regulation. Here is the structure of tryptophan operon. There are the 5 structural genes, and on the left side there is on the other side there is the regulatory regions. The repressor messenger RNA is transcripted and the repressor protein is synthesized, and if the tryptophan is in high concentration, then two molecules of tryptophan attaches to repressor and both of them binds to operator site and transcription is inhibited. But if the tryptophan level is low, then no tryptophan is available for binding with the repressor, and so no binding with the operator site and the transcription starts. And another is attenuation control. What happens if tryptophan is present? Any of the RNA polymerase escapes repression at the operator site. The transcription begins at the promoter, but prematurely terminated at the attenuator region. We see there is the attenuator element between the structural genes and regulatory region. If any polymerase, RNA polymerase escapes the binding with repressor, then it starts transcription, but it is terminated by attenuator element. And if tryptophan is absent, the attenuator region has no effect on transcription, and the tryptophan operon structural genes on the tryptophan operon are transcripted. So now we see lac operon is inducible by lactose and tryptophan operon is repressible by tryptophan. The coordination of transcription and translation in prokaryotes, that is by a stringent response, and we see the regulation in response of 7 operons of ribosomal RNA synthesis in E.coli is done by amino acid starvation or other environmental conditions regulate its response. If there is any uncharged transfer RNA, then alarmone guanosine tetraphosphate is synthesized, which is a derivative of GDP, and its elevated level inhibits the ribosomal RNA synthesis. And there are certain regulatory ribosomal proteins which regulate the transcription in the prokaryotes. And these ribosomal proteins inhibited by an excess of their own protein products. The R protein binds to ribosomal RNA with higher affinity than messenger RNA, and so they inhibit their own products. If concentration of ribosomal RNA falls, the R protein is available to bind its own messenger RNA and inhibits its translation. And this coordinated regulation keeps the synthesis of R protein in balance with the transcription of ribosomal RNA. Here we can see the operators, and the operator site is the ultimate site which have to either initiate the transcription or inhibit the transcription. All the regulatory molecules or initiating complex attaches, binds to the operator side. So finally, operator have to function. Either he has to be stopped or either he has to go on. So the transcription is regulated. Thank you. We will meet again in the next class for the gene regulation in the eukaryotes.